Good afternoon. Thomas Cordes here, the representative of Brussels, was predicting that very soon there will be a Bulgarian participant in the Human Brain Project. But as somebody from Greek origin, he should know there is a Trojan horse, and I'm not Macedonian, I'm not Polish, I'm Bulgarian, and I'm happy to present you here part of our work in the Human Brain Project, namely yet another platform, the Medical Informatics platform. And as he suggested, time is for new people, for young blood to come, and I'll present you what me and my colleagues were doing in the previous stages of the Human Brain Project, and in the current phase, I'll be happy to step aside and look from and admire from aside the amazing work of the Human Brain Project. Okay, Medical Informatics platform, why is it so different than the other platforms? Well, because we are facing a lot of challenges in clinics. As I mentioned, I'm a neurologist. I see patients with dementia, I see patients with epilepsy, I see patients with stroke, and we do not truly understand the majority of these disorders. You know, there is enough evidence when a clinical diagnosis, particularly of neurodegenerative disease, is put forward. Afterwards, the patient eventually dies. We do the histology evaluation, and it turns out to be a completely different disease. That's why we're asking ourselves, what are we treating? Then, of course, drug development is very expensive and slow. You heard a month ago, it was a company called Pfizer that put out their research and development part from about neurodegenerative disorders. And of course, the main point, what we're talking about, is data. And we know data is expensive, data can win elections, data can make countries jump out of the European Union. That's one of the dilemmas. That means when you're asking how can we put this and that data, you have to think about this data as a potential value. Then the question comes, whom belongs this data? Is it belonging to the institution that is acquiring the data, or is it belonging to us as taxpayers and payers of our insurances that are going somewhere to get a brain scan, for example, for diagnostic purposes? I think the second one is more valid. And the third point is, of course, a double-edged sword. It's the so-called GDPR, the General Data Protection Regulation, which we see again in the example of Facebook, Cambridge Analytica, etc., is something good to make. Indeed, there have to be regulations, and Europe is really at the forefront about this. But data sharing, that in our scientific world is one of the most noble words, suddenly becomes a non-word. All of the titles of the press releases around Cambridge Analytica and so on were data sharing, how bad it is. That means we have to cope with this. Particularly here, if we're talking about medical informatics and data which is concerning our own private, intimate sphere of health, how are we going to do this? Well, we have, I think, a certain answer we have provided, and let's see how this rolls out. We have a solution, I think, about the data and accessibility I'll be talking about, but here the idea is very simple. The idea is to have a browser-based, as the majority of here virtual and non-virtual platforms you saw. Through the browser you access certain amount of data that you shape according to your query in order to define a clinical hypothesis. Because, of course, even in our world of research, we sometimes start collecting data and suddenly we ask ourselves, why are we collecting this data? What is it good for? That means, first of all, of course, it's good to know what kind of data is available. That means I'll try to demonstrate you a little bit of descriptive statistics, how many patients are there, what kind of patients are there, male, female, etc. And of course, when we have put forward this neurobiological question, that means we have a hypothesis, we have to test it. And the Medical Informatics platform also offers this possibility that we test the hypothesis with, of course, statistical means. We are clinicians, but nevertheless, we count on statistics, not on our experience in human eyes, because they are the majority of times wrong. Once we have tested our hypothesis, we validate it. Victor was talking about this. This is very important, be it on retrospective data or, of course, prospective data, much more interesting and valid. This is the medical informatics platform. Our system tries not to suck data from different hospitals, but much more to federate data. Again, I'll be explaining this in detail. And we think that this opens doors to in-depth understanding of the brain. Among the 7 hospitals that are participating, also, unfortunately, not from Sofia, I'm from Sofia, but from Plovdiv, we have participants from the medical university, and I think this is a venue that eventually will also continue with Varna. Good. This is a very busy slide. You don't have to look at, but the idea is indeed to gather all data at different levels of observation, starting from genetics, synaptic, cellular, systems, up to the brain, and then its interaction with the environment. Even socioeconomic data and so on. Somebody was putting the question forward, what's the amount of data? Well, it is big data. If we're talking about biological data, it's petabytes. And if we're talking about the variables which are much more coded as numbers, we're talking about gigabytes. Of course, we need lots of data. Why do we need a lot of data? Artificial intelligence is a buzzword at the moment, although it's just summarizing concepts that we know about since decades, machine learning techniques and so on. And you know, in order to make more and more accurate models, you need more data. And what else do you need? You need also big computers. And of course, we're very proud to be in such a union like the European Union, but let's be very clear, the European Union is far behind the other competitors. If we're counting supercomputers, China has more than 200. The United States, more than 140. Germany, 20. France, 18. The UK, building the 15th. Bulgaria, I don't know. That means, what are we talking about? That means neither do we have a concept how to gather big data, besides, of course, rigid good regulations like the GDPR, nor we have the capacity to number crunch this data in a proper way. Good. The idea is now having all this data from hospital research bases and so on at the various levels with features extracted. That means not now looking at some molecules and brains and so on, but really something at the abstract level where machines can work with it. We can start indeed looking at this big data, defining homogeneous subgroups and so on, something what we need, redefining medicine, because this is our problem in medicine. We're working still with concepts from the last century when the grandmasters, for example, of neurology from France, UK, so 1, 2, 3, 4 individuals with a similar phenotype and say, well, this is, I think, the disease of Draganski. Well, fair enough. There was a certain foresight, there was a certain understanding of the clinical phenomena, but now with all the information coming from genetics, we know that one and the same gene can cause different clinical pictures and vice versa. Very similar clinical phenotype can come from different genetic and pathophysiological backgrounds. Good. And of course, this is our aim and my personal, as a clinician, to turn back this knowledge to the patients, indeed come up with this predictive medicine at the age of 46 to say, okay, he will have Parkinson's, Alzheimer's disease, or nothing, or even personalized medicine, something what we need badly. Good. What are the goals of the medical informatics platform? We have built, that's why I was saying I was active in this previous period up to now. From now on, there are other people taking the flame ahead to build the tools to federate clinical data, which are currently locked in the archives of hospitals for legal reasons and so on, and we are not benefiting as researchers from this data. Of course, we have to somehow please our partners, the hospitals, because I said data has its value, and we have to provide incentive. Unfortunately, the 1 billion is not exactly 1 billion for the Human Brain Project, it's less, but let's not talk about numbers. The Human Brain Project is not collecting data, it's not distributing money, it's about, indeed, finding mutual interest into certain activities, and this is how it should work, I think. But nevertheless, we have to find a sweet spot how to insensitize the hospitals. And what we are working on very hardly, it is to develop tools to extract these features from biological data, because this is very, very difficult. Hospital data are dirty data, there are a lot of missing data, there are a lot of errors, and this is where we have to talk about and develop tools about ontology. Diagnoses are given according to different systems, although we have DSM-4, DSM-5, etc. Sometimes they're in Cyrillic, in Latin, in one language, in another. How should this work? But again, there are bright brains behind it that have answers for all these questions. This is one of the many versions of the medical informatics platform that I'll show you. The smart idea here, not to really touch on this very sensitive topic of data privacy, is actually not to expose any raw data to the end user, who is this number 4. The end user should work like through a system similar to Google, ask a query. I need all 60-year-old left-handers across Europe, male, for some reason, and then it will go down to all the different participators of the consortium or of the platform, hospitals, research databases, and so on, will enter there the query, but already locally in all these hospitals and research bases, there will be a computer which is number crunching and organizing the data in such a way that as a metadata, that means my question will be about the 60-year-old left-handed 60-year-old male across the different centers. It will make a mean or average in all of these different centers, and this metadata will be presented at the higher level. Of course, there are certain also statistical methods that they have to be applied, because each center has different numbers of participators, etc., etc. And the end user will work only on this metadata. That means we are creating a summary of each center's data without the necessity to represent this data to the interactive level of the end user. There are different questions possible. One of our collaborators is Florence Pasquier from Lille, and she is interested in dementia. And for example, some of her questions are, what are the factors influencing disease progression in Alzheimer's disease? For example, with my query about the 60-year-old left-handed male, I'll be looking, are Bulgarian left-handed male more prone to Alzheimer's than the ones in Switzerland? Is it because Switzerland is much more rich, or is it because the air pollution in Sofia is so bad, etc., etc.? All these questions. Or what is the contribution of vascular pathology to Alzheimer's disease and its progression? Because we know they are also intimately intertwined. What is the differential diagnosis between different phenotypes with the clinical picture of dementia? And so on and so on. Other type of user is the radiologist, Karl Egger from Freiburg. He's asking, can I then as a radiologist benefit from all this access to different hospitals, which although they have different scanners, different types of imaging protocols and so on, you, with your tools in the medical informatics platform, can extract these features, like local volumes of the brain or everything what Victor was explaining before, in a more simplistic way, in order then to generate knowledge based on much more distributed and, in terms of also numbers, bigger data. Or how, as a radiologist, rather than relying on my eyes, I can use these automated tools that are existing in the medical informatics platform that, from a complex image like an MRI scan of the brain, can really pixel by pixel estimate volumes, thickness of the cortex, etc. And of course, more interesting, because we are trying to broaden our scope, not only look at structures of the brain, etc., but how are they linked to genetic data. Catherine was talking about the Allen Brain Atlas, great tool which is giving us already, although just in 6 humans, the spatial distribution of genetic expression, etc., to really answer a certain question between the link of between genes and nature and nurture. Good. Now, everybody said, don't do it live, but I'll risk it. I wanted just very quickly to show you the interface. Well, this is the Human Brain Project. Of course, I'm not going to enter into the analytical part. I just wanted to represent you the exploratory part. We created these bubbles, which are giving you the possibility either to zoom in different regions of the brain, roughly 136, or exactly, and also some clinical data like cerebral spinal fluid biomarkers of Alzheimer's disease, some clinical scores like in Parkinson's disease, UPDRS, the mini-mental, the MoCA, genetics, some SNPs, etc. How does it work? I'm interested in a particular variable, for example, hippocampus, interesting structure, neurogenesis, atrophy in dementia and aging. And then already online, sorry, but I couldn't squeeze the screen. We have here live the histogram of volumes. It's a normal distributed, of course, data set. Here what you do not see, the different colors are the different cohorts that we have from the different hospitals and also from the big research databases of the Alzheimer's Disease Neuroimaging Initiative. You can look, well, we had to put actually sex, not gender. Sorry for this. How does it distribute among female in black and male in blue? We can look at the distribution by age. You can imagine that here the x-axis is the age, and of course here you have the number of participants. This is what I was talking about, this descriptive statistics part. That means you can go through the features of the brain, you can go through different genetic markers and so on, orient yourself, what are the numbers, what are the diseases, etc., etc. Very nice, much more powerful, I think, is the analytical engine behind, which then, accordingly to your query, is not only giving you the possibility to make univariate analysis, what everybody can do, t-tests and regression analysis and so on, but we have a very powerful multivariate analytical tool, which again gives you then further insight that you can extract from to again return to the data, postulate new hypotheses, etc., etc. I think it's a good work that we did up to now. Now the promised second type of representation of our architecture, because I have to admit we failed in really creating this incentive for the hospitals, and that's why we decided to split a little bit the development of the platform virtually into a local form of the medical informatics platform. That's why we call it here MIP local, and the federated one, which has this noble principle of federating data. The idea of the MIP local is to create an incentive for the clinicians that can use locally the powerful tool of feature extraction and data analytics for their own patients in the hospital. Because otherwise, if you would be, let's say, opening this version to the world, then of course you have problems with data privacy, etc. That means just a tiny encapsulated version, but with all tools and features of the medical informatics platform, can be used locally, can also benefit from all the knowledge that you can create from the existing data. Then you have your own single patient, upload his or her data, number crunch it, and then do this predictive diagnosis. Is it more probable that according to structure, certain biological and other data, this individual has much more this type of dementia or other type of dementia, for example. And this is a little bit this modern vocabulary. Data capture is, of course, the principle of getting the data, which is residing, for example, imaging data in some PACS system of radiologists, etc. Everything is solved. The data factory is this engine which is crunching all the data and organizing it. Algorithm factory is this knowledge about statistical methods that in a very intuitive way you can use, clicking this and there. And of course, there is the web modeling and visualization, which gives you, let's say, the pleasing pictures that also Victor was showing. This is the MIP local, the MIP federated. You can imagine this times 10 or 7 at the moment, the 7 hospitals participating, where you have the same principle, but then the data stays locally, and as I said, according to your query, all these queries are descending to the different participating centers, extracting the necessary metadata, and giving you the opportunity to do your meta-analysis with using the same analytical methods that you'll be using also locally. I'm going now quickly to explain you what's the work behind it. If you want, of course, you're more than welcome to explore the platform. It necessitates a password that you can obtain via the Human Brain Project website. I was talking about feature extraction, and in a simplistic way, we can say we have three-dimensional imaging data of the brain. We know already for many years how to split with accuracy of sub-millimetric dimension the different components simplistically of the brain, gray matter from white matter from CSF. Of course, I was saying this is a challenge in medical data in order to save space and time. Data is either not acquired at really minimal space between the different slices, or it has a lot of artifacts because patients move, etc. And this is one of our collaborators, John Ashburner, professor at the UCL, who is working on this data, how to combine bad quality data to exactly extract these features about volume of gray matter, white matter, etc. This is the general principle how I'm now diving into the brain imaging part. What kind of methods are we using? Also the platform. We can either for each pixel, what in the three-dimensional space we call voxel, we can estimate based on intensity and also a certain position in space. It's a Bayesian framework, that means probabilistic. The probability being gray or white matter. We can also estimate the so-called cortical thickness by detecting automatically the boundary between white matter, and CSF. And at the end of the day, what I was saying, we're not radiologists, just eyeballing images and saying, according to my experience, he's a little bit less, he's a bit more. We do statistics. That's very important. For us, the MRI machine is not a diagnostic tool. For us, the MRI machine is a research tool which is delivering data that we submit to statistical analysis. We did some work on subcortical structures. Katrin was talking about the subthalamic nucleus, which is a very important target for this electrical stimulation through pacemaker in Parkinson's disease, but also other parts of the basal ganglia, like the pallidum and so on, are targeting other movement disorders like dystonia. For MRI these areas of the brain are very difficult because for some unknown reason, they have lots of iron, and the more iron, the less you see there. We have developed also certain MRI techniques where we can acknowledge this contribution of iron, but then I was talking about Bayesian framework. That means in order to separate gray and white matter, we want to benefit from some prior knowledge. And this prior knowledge in our case are certain, we call them tissue probability maps. And we work on this in the framework of the Human Brain Project in order to create such tissue priors for the basal ganglia. We, of course, wanted to be modern. That's why we did a little bit of crowdsourcing. We created a web-based tool where the images of 100 individuals in a good age range span were presented to students and other volunteers. And these students had manually to delineate these structures because there are no reliable tissue priors existing for these structures. And of course, somewhere, unfortunately, you have to start with your hands. In this way, we created these new tissue priors. And if you see here, this is the result of delineation of gray matter with the old tissue priors and with the new ones. And you see very clearly how here in this bottom row, we are benefiting from this tissue priors to detect the substantia nigra, the pallidum, also here, and the thalamus. Structures which are missing here using the old ones. Small work, but important. Good, of course, we use statistics here. All these colored blobs are just statistical maps that we see the difference between the old and the new way how we do it. This is a very, really superficial summary what we did up to now in the medical informatics platform, because you ask yourself, well, developing tools platform, but we did also indeed some empirical work. We worked on Alzheimer's disease. We created data-derived indices of symptom severity, because sometimes if you are from genetics or other background, we are not interested in all these pixels and type of analysis that we are so fond of with and about, but you're interested just getting one number per brain, an index of its pathology or disease severity. And this is something that we empirically showed for the case of Alzheimer's disease, looking at some more, let's say, questionable clinical pictures like post-traumatic stress disorders. This is work from colleagues in Israel. Again, Alzheimer's disease, disease progression, again for neurodegenerative diseases, and so on and so on. Then we realized, actually, in order to incentivize our colleagues from hospitals, one of the most important questions is to create this baseline about aging. Because most of the neurodegenerative disorders, they start with older age, but of course we have a 60-year-old one with Parkinson's disease, 90-year-old with Alzheimer's disease. That means we have to ask ourselves, what is actually the trajectory of normal aging in order then to reliably describe the pathology of one or another disease. And of course, I'll be talking a little bit more about our work on aging that we did in more than 1,500 controls from Switzerland. We also developed our own tools, which are complementary to the ones that Katrin was presenting, how to link the space of the brain, of MRI and so on analysis, with the very famous Allen Brain Atlas, with the distribution, spatial distribution of gene expression across the brain. Well, one of the main questions when we talk about MRI is indeed bridging the scales. Because on the one hand side, we want to understand the biological underpinnings of all these disorders, but we cannot open the brain. MRI is giving us certain abilities to look into it, but up to now they've been very descriptive and very far away from underlying biology. Victor was talking about, and Katrin and all the others, we are very much skilled into representing certain values from inside the brain or outside the brain, on the surface, on these pixels or voxels. But this is exactly what we want to do, to bridge these scales, because with MRI, we can study individuals in vivo we can study many. If you have one monkey brain or one rat or mouse brain, you are going to study just a small, tiny part of it, and you cannot study thousands. UK Biobank aiming at 100,000 human brains. Imagine this into the non-human primate or rodent work, impossible, too expensive. That means here we want to bridge the problem of spatial resolution, but we are gaining then data from a whole population. And of course, what I was talking about, at least in my lab, we're working a lot on getting closer to biology. For example, one of the very basic principles, maybe for those of you who are in the brain business, is the notion of the so-called G ratio, the ratio between the diameter of the axon and the thickness of the myelin sheet around. This has to do with basic principles of physiology, like conductance, velocity, and so on. And now, of course, we don't have the spatial resolution of 5 micrometers or so on to delineate a single axon, to measure these features like you do on electron microscopy, there, the picture on the right. But using a biophysical model, information about the axonal density on orientation, the technique that Victor was explaining so beautifully with these colorful spaghetti pictures, but additionally also measuring quantitatively the myelin in these very same pixels of voxels, we can, across the whole brain, estimate this G ratio. And again, things are starting to become very interesting. This is what we are doing. I was talking about the studies of aging we have been doing on these 1,500 individuals. Each point in these graphs is an individual. We see, unfortunately, the majority of brain structure, be it gray matter, white matter, in terms of volume, the established techniques, they go down. If we look at the white matter, which was a little bit of terra incognita up to now, things are looking slightly different, giving us hope, the middle-aged ones. Myelin is peaking in terms of reorganization in white matter at the age of 40, even 50. Unfortunately, afterwards, there is this inevitable decline. You can describe white matter in different ways. Again, diffusion tractography, these spaghetti-like connections of the brain, but you can also, Victor was explaining this, reduce the features and really present the so-called connectome, the most stable and reliable connections across the whole brain. You can do it in an abstract way as connectivity matrices and so on. This is what indeed we did for the Human Brain Project across major tracts in the brain that you here see beautifully on the left-hand side. We were measuring the different features in the different graphs, be it derived from myelin or from diffusion-weighted imaging or this concept of G ratio. You see when exactly are the peaks of these metrics. And you see certain measures like the measure of myelin, which is the uppermost on the left, MT and R1, they're showing very narrow peaks. That means in a population here of roughly 1,000 individuals, we don't have a major variability. Around the age of 40, the majority of tracts are peaking. Of course, it's interesting because it's showing also certain metrics like fraction anisotropy. This is the third from above on the left-hand side. There is a huge variability. The question is the model behind that stable? Why for the very same tracks? This is giving us a different variability, although fractional anisotropy is describing the orientation of fibers rather than myelination, etc. Of course, we have to ask ourselves, what are the patterns of aging in terms of linearity and non-linearity? There are statistical ways how you can test this if you have enough data. And indeed, we show for the most of the white matter tracts, we have this beautiful quadratic distribution of age, inverted U-shape, development up to the age of 46, and then down. And these are the estimations of the different trajectories across different tracts. Beautiful pictures. Here the gray areas are, of course, the standard deviations around the mean. And G ratio, you see, makes completely different U-shape trajectory, which has its biological reason, but we're not going to discuss this now. Then we look, of course, how is it different between male and female? No. Is it different between left and right? No. The spatial resolution has an impact, because I said clinical data is most heterogeneous, but this was indeed a major difference. But nevertheless, the results are consistent and interesting, and I think it would be a shame not to use this in the future in the framework of the Human Brain Project. And these beautiful images are just a summary of what I was talking about across the different tracts, how age changes these tracts, and we see what up to now the white matter was much more uniform, ambiguous structure, suddenly starts making sense, and you see a differential speed and trajectory of white matter changes related to healthy aging in the different parts of the white matter, the different tracts, and also the G ratio. This ratio between axons and the myelin sheath is also very different, which makes sense. Of course, the corticospinal tract, all these cables running from the sensory motor cortex down and the other way up, it has to really lead information with a high speed. That's why we need a lot of myelin, etc., early myelinating, and that's why we have a quite different here G ratio than compared to the other tracts. Very simplistic way, of course, but this was pleasing my colleagues clinicians. This is just an example of the MIP local, where individuals, these are very tiny dots, you cannot see them, but this was just one patient with a certain form of dementia, and this is how this very patient performed for different parts of the brain, either in terms of volume or myelin measure or iron, as compared to this trajectory of healthy aging. This is what I wanted to show you. That was the reason why we were using so much data, creating these trajectories, because this is what matters for a clinician, to give an answer to the patient. Well, you unfortunately in this part of the brain perform above or below certain standard deviations, certain, let's say, measure of normality. That means your brain is here and there abnormal. That's why I think you have this and that disorder. Good. Then, of course, we love atlases, but we know that atlases have their problems with when we're talking about brain atlases, because they're putting a major restriction that all functional areas are uniform across us, and that's not true. And therefore we tried from this multimodal data, talking about intracortical myelin, iron, etc., to create something data-driven. And when we mix the brains of, again, these 1,000 individuals, which are sensitive to different type of tissue properties, we see this patchy distribution of tissue properties. And indeed, when one comes into detail, it matches very well a famous paper by Van Essen and colleagues from a few years ago, that they show how actually this Brodmann or other type of architectonic-based mapping of the brain is not true. We do not have sharp borders between the different areas. It's much more gradients, but there, Katrin and her colleagues in Jülich are much more at the forefront of this type of description of the brain, but we were pleased also to confirm these results also with this structural data. Of course, we see a certain gradient, particularly driven by the myelin, if we go from the surface of the brain down, because we know that the lower cortical layers have more myelin, etc. Good. Let's make some publicity at the end. What makes us unique? Well, I think that we have developed an outstanding, very robust platform for neuroscientists and clinicians, epidemiologists, pharmacologists, etc. We have also put all the novel concept in terms of brain imaging, brain data processing, feature extraction and analysis inside. I think we can use this platform to bridge, indeed, different scales. Katrin was talking about, Victor was talking about cells, genes, structures, diseases, etc. And I think this is a powerful tool. There are a lot of demos here on YouTube. Of course, you can obtain this information from me, from my colleagues here from the Educational Bureau, and just let yourself informed by all this modern media. Good. What else did we achieve? Well, I think we came a little bit closer towards standardization of clinical and research data sets. We are helped by colleagues in Greece who are specialists in ontology in terms of creating certain schemes where we can put the different languages, formats, lab results, and so on, that we can really talk the same language. And most importantly, that the computers understand the data that we are entering. We have provided each of these hospitals and centers a software package, which is installed remotely, which is stable, which contains the best and the newest of brain analysis and data handling software. And I think this adjusted model of brain aging based on lots of interesting data, not only descriptive, but tapping into the biology of the brain is also something important. And of course, we have our small contributions towards giving at least European level certain guidelines for diagnosing and treating certain neurodegenerative disorders. Well, this is absolutely the same, just a few more pictures, nothing important. This is an important slide for me because it actually closes the circle that we are trying all the time to close with the type of research, what Victor is doing, what we are doing. Actually, scientists are indeed ahead of what happens in clinics, because I'm in these two worlds, I can tell you indeed. In clinics, for good reasons, but also sometimes for unknown reasons, research advances are not coming that fast, and we have to close this loop through indeed getting the help of data analysts and statisticians to really extract these features. I don't have to be a master in neuroimaging or whatever other technique in order then, based on this platform, to make use already of data which other specialists have extracted for me. I'm a geneticist, but I'm interested in the link between behavior, brain, and so on, but already there in a certain level of abstraction, I have certain type of information that I can use, and I think this is great. And then, of course, clinicians can make use of this information, giving back this knowledge to the patients, who are then inspiring scientists to further develop tools and techniques, and then we are going many times through this cycle until we get a better world. Good. We're very active on various forums, etc. That's the whole timeline for the last few years. Nothing most important than presenting here. And of course, at the end of the day, this is a work of many people. I had my small contribution to it. Lots of hospitals, lots of countries are represented. Bulgaria is still not on the map, but indeed with Plovdiv, again, I told you, hopefully also Varna soon will be back on the map. With this, I thank you very much, and now you have time to go for lunch.